Hello my Access to HE fledglings. Today we'll be talking about muscular contraction, and hopefully by the end of this you'll be able to explain how muscles contract, along with possibly understanding why a few diseases, musculoskeletal diseases occur, and also possibly explain how some drugs work. Now this is about the fourth time I've tried to record this video, and so if I'm getting a bit impatient you might be able to tell in my voice, but I'm going to be as sarcastic as ever. We have to do 6 or 7 things. If you're interested in only a pass, if you're only interested in the bog standard basic understanding, you will only need to go for the first 3 order of things. If you want distinctions, or if you're bothered about this topic and you want to understand how a muscle works, then you will need to know all about the 7 things that I've got for you. To start with then, the big picture, skeletal muscle is really under our voluntary control, and we require a brain really to be able to stimulate them, mainly. And so what happens is that we send a message down something called a motor neuron, and that generally is a nerve that is attached to a skeletal muscle, and it's that muscle that will help us to interact with the environment. Ever since we've been a baby, we've been training our mind and our bodies to be able to interact with the environment. So the information or the stimulus has to come from our brain. It travels down the spinal column and then out into the body, and it's where the nerve attaches to the muscle cell that's going to cause the muscular contraction. So without further ado, we need to be able to understand the structure of muscle, and then we can use those landmarks to be able to talk or describe about how that muscular contraction occurs. And in each slide we're going to get more and more in depth, but hopefully they follow one another. Stop it, rewind it, play it again to your heart's content until you're confident in grasping the knowledge. Now, a muscle often is attached to a bone. So here we're going to have a bone. It's not a very good looking bone, but it is a bone nonetheless. And that bone, as we know, has a periosteum around it, the double layered membrane, which is the site of attachment, and it's at that point in which we get the muscle attached. Now I'm going to draw the muscle here, which is cut across its section, so it's a bit like a joint of beef that's cut across the section. Now if I weren't going to cut that muscle, we'd see something like this, like that, and we're just going to sort of cut that section there with there. We're going to cross section of that muscle. I hope you get the picture. If you don't, it's tough. No, it really isn't tough. I'm hoping you get it. And to draw the structure of a muscle, all we really need to do is basically, like a telescope, pull out 3 or 4 sections that are going to help us describe the structure of muscle. So let's draw that, and that. So like I said, really, we've just pulled out one section from each layer of muscle. So here, for example, I've just pulled out one of these sections here, and it just happens to be this section that I've pulled out. And then of this section, I've pulled out this section here, and again, we have lots of different subsections here. Right, I'm going to start at the most important place for us in terms of muscular contraction, which is this section here. And this section is the cellular level of a muscle, and we're going to call that a myofiber or a muscle cell. And a muscle cell has exactly the same features as any other cell. It has a cell membrane, it has mitochondria, it has a nucleus, or multiple nucleus in this case. It has cytoplasm and rough endoplasmic reticulum, smooth endoplasmic reticulum, that has all that machinery. Now, what we do need to know is that the cell membrane of a muscle fibre has a special name called the sarcolemma. It's made of phospholipids just like any other cell membrane, but the muscle fibre or the muscle cell's cell membrane has a special name called the sarcolemma. Now, if you want to envisage what a muscle fibre looks like, if you got a load of drinking straws and held them together in your hand, and then wrapped those drinking straws together with some cling film, that's the kind of structure you would see in terms of a muscle cell or a myofibre. The cling film would represent the sarcolemma, and the drinking straws inside would represent these structures here, which are poking out, called myofibrils or myofibrils. Depends where you live. Myofibrils. And myofibrils are the functional unit of skeletal muscle, and they allow the muscle to squeeze and contract. So myofibrils often described as being stripy. And each one of these stripes is called a sarcomere. And we're going to look at those in much more detail in a second. Just a few extra labels which I'll give you, and this is connective tissue. So we have a kind of a sheath around the outside of a muscle. That would be the epimysium, epiperi, and then the the connective tissue, which I'll draw in green here, around each fascicle, around each sort of section of this layer, is called the perimysium. Might as well write mycium on it just so that you know how it's spelled, mycium. And then the connective tissue around each muscle fibre or myofibre or muscle cell is called the endomysium, and it's just connective tissue. It's just the different names for the different levels of connective tissue which separate each section and subsection, etc. But that is not the main event, because what we need to know is the structure of a sarcomere, and we need to know the labels of a sarcomere so that we can accurately describe how muscles contract. Well, sarcomeres are defined by Z lines. These are the zigzag lines on either side of a sarcomere. So this here is one sarcomere, and it's identified by one stripe of the myofibril. Extending horizontally in the diagram, but remind yourself that this is a 360-degree structure, and therefore it's sort of circular, and it's a section of the rod of the myofibril, which is hard to understand until you understand it, if that makes sense. There are two contractile proteins within muscle. The first, which I've drawn in blue here, which is actin. And the second, which I'm going to draw in red, is called myosin. And we have myosin running in between the actin filaments. So they're both proteins, and we describe them as filaments because the protein's in like a filament form, the kind of long and strandy. This red protein is myosin. So if you ever ask what are the two major proteins involved in muscular contraction, you can say actin and myosin. Actin is thinner, myosin is thicker, so we often call it thick myosin and thin actin, just so that you know. Now, that is basically what a sarcomere is. We just have to add on landmarks to describe these structures, and they're actually more obvious than you might want to imagine. These black zigzaggy lines are called Z lines. We can imagine a midway or imaginary halfway line. That would be called an M line. Common sense. I'm going to do this in green. The gap between the actin filaments, between the blue lines, now I like to draw it like this because that green line there reminds me of a H. And this gap here, this gap, or the distance between the actin filaments, is called the H zone. Ooh, the H zone. The width of the myosin, let me draw this in, I'm going to draw this in blue. So here to here, that distance there, that distance, the width of the myosin is called the A band. Now do that because it looks to me like I can draw an A on top. I draw an A, this diagonal line here reminds me that I can draw an A. Now if we imagine there's a sarcomere next door, so if you look at the myofibril in the top right of the screen there, you can see that the sarcomeres are neighbouring each other, one and another and another and another and so forth. So we can see that there's going to be actin on this side and there's going to be myosin on this side. The last label that we need to put on there is the I band, and I draw the I band like this. So if I can draw the I band there, I can draw a big I, and basically it's the distance between where the myosin finishes on this sarcomere and the myosin finishes on this sarcomere. So it's that distance there, and I draw a big I to represent that because it's called the I band. So if I can draw an I to represent the I band, it helps me remember it, the I band. Okay, we've got all the information and labels on there to do a good job of describing how muscles contract. So let's put that into some kind of detail. Let's draw the sarcomere again, just quickly. Got the Z lines, you've got the actin filaments poking out, and you've got the myosin filaments in the middle. You've got the H zone, you've got the A band, and you've got the I band, and you've got the M line. So when a muscle contracts, and this is really important, the actin slides together on each sarcomere. So you can see that the arrows that I've put on there, the actin slides together, and in doing so, the Z lines move that way, the Z line moves that way, and it's because this myosin here grabs hold of the actin and moves it that way. This actin, the myosin grabs hold of the actin and moves it that way. This myosin grabs hold of that actin and moves it that way, and so forth. So what happens is you get this sort of interlock, you get this actin sliding towards the middle. And if every sarcomere on a myofibril, here's my myofibril, if remember there the Z lines on a myofibril, if every Z line moves together on a myofibril, you're going to get contraction. And if every myofibril contracts and every muscle fiber contracts, then you're going to get muscular contraction. So we call that, oh, well actually what we say is that actin slides over myosin shortening the length of each sarcomere. This is called Huxley's sliding filament theory. Forgive my handwriting, I'm rushing big style. So when a muscle contracts, actin slides over myosin, shortening the length of each sarcomere, which is called Huxley's sliding filament theory, after the guy who investigated it, proposed it. And what we can use is our 4 labels to describe what happens: our Z line, our H zone, our I band, and our A band. So the Z lines, it's obvious. The Z lines move towards the M line. when a muscle contracts, concentrically anyway. The H zone reduces or disappears even. There is no H zone sometimes if a muscle's contracted fully, and actually overlaps anyway. The I band also reduces because the Z line moves this way closer to the M line, and if you imagine the other myosin filaments doing that too, you'll notice that they do squeeze together. And it's important that you know the A band stays the same width. The A band stays the same width, so the myosin heads and the myosin filament, this stays the same. It just grabs hold of the actin and moves it closer to the M line. Okay, that gives you the fundamental description of muscular contraction and the Huxley sliding filament theory. What it doesn't explain is how the body sends a message to the muscle and makes it contract. So first of all, what we've got to describe is the nervous stimulation, and here we've got the end of a neuron called the axon terminal. This is the end of a motor neuron, and the signal arrives at the end of a motor neuron, which triggers the influx of calcium. The influx of calcium triggers a snare protein, this thing in red, to pull on vesicles containing this green stuff, which is a neurotransmitter called acetylcholine or acetylcholine. You'll have heard of the neurotransmitters called serotonin, dopamine, etc. In muscular contraction, that's called acetylcholine. Well, that acetylcholine is released into the gap via exocytosis as a result of this calcium flooding in. The acetylcholine diffuses across what's called the synaptic cleft, which is the gap between the axon terminal and the sarcolemma, which is the name for the membrane of a muscle fiber or a muscle cell. The acetylcholine travels across or diffuses across the synaptic cleft and binds to receptor proteins on the sarcolemma. And that's what we mean by a neurotransmitter. A neurotransmitter is released and it transmits a signal from a nerve to something else. In this case it's a muscle, but often it's another nerve, and that's what a neurotransmitter does. Upon releasing, or upon receiving, should I say, the neurotransmitter on the membrane of the muscle cell, this triggers the myofiber, or muscle cell, to kick off its own impulse, which spreads along the sarcolemma. That's actually meant to be a lightning bolt. It's the worst lightning bolt I think I've ever drawn. Anyway, this signal travels across the sarcolemma and disappears into the muscle, deeper into the muscle, via tubules called T-tubules. The signal spreads into it. And there it meets sacs full of calcium. These sacs are called the sarcoplasmic reticulum and are extremely important for the next slide. If we look closely at myosin, myosin has heads. It has heads on it, and it's quite a relatively straightforward molecule in comparison to actin. Actin looks like two strands of pearls on a necklace that are wound together. So these are actin molecules. And if I looked at one actin molecule, I would see that it has a kind of binding site on it. So this kind of notch. And when muscles contract, this myosin head binds into that notch and pulls it. Now, the notches on the actin molecules I'm going to draw with black dots very quickly. Because like I said, I've done this 4 times now and my microphone's let me down each time. Now, at rest, when these binding sites are exposed, as they are now, the myosin heads want to bind into this binding site, into this notch that we've drawn up here. But when a muscle is at rest, we have another protein on actin that covers the binding sites. It covers the binding sites on actin. So I know this muscle must be at rest. Now this protector of the binding sites, this is a binding site on actin. This green line, this protector, is called tropomyosin. Tropomyosin. And we also have another protein which I like to think is kind of a ribena also on actin too. So how does a muscle contract, I hear you say? Well, this links to our T-tubule coming down here and also our sarcoplasmic reticulum, sac of calcium, here. Here's the calcium in the sacs. So as the signal transmits down the T-tubule, it triggers these voltage gated proteins to open. And when they open, they release calcium into the myoplasm, into the cytoplasm of a muscle cell. This calcium binds to this blueberry, which is called troponin, this other protein that's on actin. And when calcium binds to troponin, this moves tropomyosin away from the binding sites. So tropomyosin changes shape and exposes these binding sites. And when these binding sites are exposed, that's when the myosin head can grab hold of that binding site and form a cross bridge. Now the cross bridge is basically where the myosin head binds into this binding site, which is what I've drawn up onto this, on this top right. Okay, on to the next one, which is the cross bridge cycle. Where do we get the energy from? Well, here's a myosin head, and what has to happen if you're firing a catapult is you have to invest some energy. So if I was going to fire something forward with this myosin head, I would draw it back into its cocked position, and I would fire it, twang, which is called its power stroke. No sniggers, please. Power stroke. So what happens is we get our energy from a molecule called ATP. And when ATP from the mitochondria binds onto the head of the myosin heads, we break this down into ADP and P. If you're interested, ask me in class to explain that. But what that does, it provides the energy to do the cocking, to cock the myosin head into that position. Energy. That's what pulls the myosin head back. The fact that we've gone from ATP to ADP and P. And at that point, we form the cross bridge. And here's your actin. I'm not going to draw it in massive detail. And so what happens is when that's been hydrolyzed, when that ATP has been broken down into its ADP&P to provide the energy to pull that myosin head back in the cock position. This ADP&P come off the myosin head, which causes the myosin head to go forward into a power stroke. And if you imagine this is the Z line and here's the M line, then that's going to make the whole actin move towards the M line and the Z line to move towards the M line. And it's this cross bridge cycle and it's this power stroke of the myosin heads that causes the shortening of each sarcomere. And that is how muscles contract in 25 minutes. You're going to have to replay the video at points, you're going to have to recap it, but I didn't want it to last too long because I wanted you to be able to rewind it quickly and get through the information and also understand it as best you can. We can actually put this in 10 points. So I'm going to recap it very quickly, this explanation of muscular contraction. So first of all, the signal arrives at the axon terminal. This causes the influx of calcium. Calcium causes the snare protein to pull on the vesicles filled with a neurotransmitter called acetylcholine or acetylcholine. Resultantly, that causes exocytosis of acetylcholine, which diffuses across the synaptic cleft. It binds to receptors on the sarcolemma. What am I doing? Why am I not numbering that? What an idiot. So that would have been number 2. This would have been number 3. I was in full flow then as well. So acetylcholine binds to receptors on the cell membrane of the muscle fiber or the sarcolemma. This kicks off its own nervous impulse, which travels across the sarcolemma and down into the T-tubules. As the signal travels down the T-tubules, I think it's number 4, number 5, goodness me. As the signal travels down the T-tubules, it triggers voltage-sensitive proteins to open and the sarcoplasmic reticulum to release calcium. This calcium then binds to troponin, number 6, which then attaches to troponin. The troponin shifts tropomyosin out of the way of the binding sites on actin, and the myosin heads, oh my god, it's 7, binds to the actin molecules on the binding sites, forming cross bridges. In order to make movement happen, ATP binds. I'm in a time warp. That's number 8. ATP binds onto the heads of the myosin, which is hydrolyzed into ADP&P, and that puts it into its cocked position. Finally, the ADP&P pop off or move away from the myosin heads, which causes the power strokes of the myosin heads and therefore shortens the length of each sarcomere. I hope you got that. I'm not doing it again. Good luck.